Good day, this is Professor Will, CBMD PhD. Today is Saturday, March 28, 2020. It is 1:51:16 seconds a.m. Eastern Standard Time. And this is a lecture and brief history on corona. So let's get started here. Missing link in coronavirus jump from bats to humans could be pangolins, not snakes. Date March 26, 2020. Source American Chemical Society. Summary. As scientists scramble to learn more about the SARS-CoV-2 coronavirus, two recent studies of the virus genome reached controversial conclusions, namely that snakes are intermediate hosts of the new virus, that a key coronavirus protein shares uncanny similarities with an HIV-1 protein. Now a study refutes both ideas. It suggests that scaly anteater-like animals called pangolins are the missing link for SARS-CoV-2 transmission between bats and humans. Understanding SARS-CoV-2, the virus that caused the COVID-19 pandemic, came from how it spreads is important for its control and treatment. Most experts agree that bats are a natural reservoir of SARS-CoV-2, but an intermediate host was needed for it to jump from bats to humans. A recent study that analyzed the new virus genome suggested snakes as the host despite the fact that coronaviruses are only known to infect mammals and birds. Meanwhile, an unrelated study compared the sequence of the spike protein, a key protein responsible for getting the virus into mammalian cells, of the new coronavirus to that of HIV-1, noting unexpected similarities. Although the authors withdrew this preprint manuscript after scientific criticism, it spawned rumors and conspiracy theories that the new coronavirus could have been engineered in the lab. Researchers and colleagues wanted to conduct a more careful and complete analysis of SARS-CoV-2 DNA and protein sequences to resolve these issues. Compared to the previous studies, the researchers used larger data sets and newer, more accurate bioinformatics methods and databases to analyze the SARS-CoV-2 genome. They found that, in contrast to the claim that 4 regions of the spike protein were uniquely shared between SARS-CoV-2 and HIV-1, the 4 sequence segments could be found in other viruses, including bat coronaviruses. After uncovering an error in the analysis that suggested snakes as an intermediate host, the team searched DNA and protein sequences isolated from pangolin tissue for one similar to SARS-CoV-2. The researchers identified protein sequences in sick animals, lungs that were 91% identical to the human virus proteins. Moreover, the receptor binding domain of the spike protein from the pangolin coronavirus had only 5 amino acid differences from SARS-CoV-2, compared with 19 differences between the human and bat viral proteins. This evidence points to the pangolin as the most likely intermediate host for the new coronavirus, but additional intermediate hosts could be possible, the researchers say. The authors acknowledged funding from the National Science Foundation, the National Institutes of General Medical Sciences, the National Institute of Allergy and Infectious Disease, and the Extreme Science and Engineering Discovery Environment. Modeling study estimates spread of 2019 novel coronavirus. Date January 31st, 2020. Source The Lancet. Summary. The new modeling research estimates that up to 75,800 individuals, as in the Chinese city of Wuhan, may have been infected with 2019 novel coronavirus, 2019 nCoV, as of January 25, 2020. The authors caution that given the lack of a robust and detailed timeline of records of suspended probable and confirmed cases and close contacts, the true size of the epidemic and its pandemic potential remains unclear. Not everyone who is infected with 2019 nCoV would require or seek medical attention. During the urgent demands of a rapidly expanding epidemic of a completely new virus, especially when system capacity is getting overwhelmed, some of those infected may be undercounted in the official register. The apparent discrepancy between our model estimates of 2019 nCoV infections and the actual number of confirmed cases in Wuhan could also be due to several other factors. These include that there is a time lag between infection and system onset, delays in infected persons coming to medical attention, and time taken to confirm cases by laboratory testing, which could all affect overall recording and reporting. The new estimates also suggest that multiple major Chinese cities might have already imported dozens of cases of 2019 nCoV infection from Wuhan, in numbers sufficient to initiate local epidemics. The early estimates underscore that it will likely take rapid and intermediate scale-up of substantial health control measures to prevent large epidemics in areas outside Wuhan. Further analysis suggests that if transmissibility of 2019 nCoV could be reduced. Both the growth rate and size of local epidemics in all cities across China could be reduced. If the transmissibility of 2019 nCoV is similar nationally and over time, it is possible that epidemics could be already growing in multiple major Chinese cities, with a time lag of 1 to 2 weeks behind the Wuhan break. Large cities overseas with close transport links to China could potentially also become outbreak epicenters because of the substantial spread of pre-systematic cases unless substantial public health interventions at both the population and personal levels are implemented immediately. Based on our estimates, we would strongly urge authorities worldwide that preparedness plans and mitigation interventions should be readied for quick deployment, including securing supplies of testing reagents, drugs, personal protective equipment, hospital supplies, and above all, human resources, especially in cities with close ties with Wuhan and other major Chinese cities. In the study, researchers used mathematical modeling to estimate the size of the epidemic based on officially reported 2019 nCoV case data, domestic and international travel, important example, train, air, road data. They assume that the serial interval estimate that the time it takes for infected individuals to infect other people for 2019 nCoV was the same as for severe acute respiratory syndrome SARS Table 1. The researchers also modeled potential future spread of 2019 nCoV in China, and internationally accounting for the potential impact of various public health interventions that were implemented in January 2020, including use of face masks and increased personal hygiene, and the quarantine measures introduced in Wuhan on January 23rd. The researchers estimated that in the early stages of the Wuhan outbreak from December 1st, 2019 to January 25th, 2020, each person infected with 2019 nCoV could have infected up to 2 to 3 other individuals on average, and that the epidemic doubled in size every 6.4 days. During this period, up to 75,815 individuals could have been infected in Wuhan. Additionally, estimates suggest that cases of 2019 nCoV infection may have spread from Wuhan to multiple other major Chinese cities as of January 25th. Together, these cities account for over half of all outbound international air travel from China. While the estimates suggest that the quarantine in Wuhan may not have the intended effect of completely halting the epidemic, furthermore analysis suggests that transmissibility of 2019 nCoV could be reduced by 25% in all cities nationally with expanded control efforts. Both the growth rate and size of local epidemics could be substantially reduced. Moreover, a 50% reduction in transmissibility can shift the current 2019 nCoV epidemic from one that is expanding rapidly to one that is slowly growing. It might be possible to reduce local transmissibility and contain local epidemics if substantial, even draconian measures that limit population mobility in all affected areas are immediately considered. Precisely what and how much should be done is highly contextually specific, and there is no one-size-fits-all set of prospective interventions that would be appropriate across all settings. On top of that, strategies to drastically reduce within-population contact by canceling mass gatherings, school closures, and introducing work-from-home arrangements could contain the spread of infection so that the first imported cases or even early local transmission does not result in large epidemics outside Wuhan. Authors point to several limitations of their study, including that of the accuracy of their estimates depend on their assumption about the zoonotic source of infection in Wuhan. They also highlight that the models assume travel behavior was not affected by disease status, and that all infections eventually have symptoms. So it is possible that milder cases have gone undetected, which could underestimate the size of the outbreak. Lastly, they note that their epidemic forecast was based on their intercity mobility, data from 2019, and might not reflect mobility patterns in 2020, particularly in light of the health threat posed by 2019 nCoV. Good day, this is Professor Will, CBMD PhD on infectious disease and microbiology. Hope you enjoyed listening and have a great day.